Dr. NSENGIMANA. He is the Deputy Director of the Center of Excellence in Biodiversity and Natural Resource Management. He's also a lecturer in the College of Education, and he's been doing some amazing work with ants and ant taxonomy, ant sampling, and finding new species, and also looking at how ants can be indicators of soil health and restoration. So I'm really happy that he was interested to give this talk today in our group, and like to welcome you, Venus, to take over the screen and tell us about your work. Thank you.
Thank you, Beth. Thank you, colleagues, for attending this seminar for the presentation. Allow me to share with you the screen. Can you see my screen?
Yes.
Okay, thank you very much. The presentation of today is focusing on the status of soil physicochemical properties in savanna and forest restored using ecosystem-based adaptation approach in eastern Rwanda. So this is a component of a big project that is focusing on the use of soil erythropods of bioindicators in the restoration activities using ecosystem-based adaptation. So we did not want to focus on soil erythropods only. We also judged good to look at properly at the soil physical properties, and this is the component focusing on the soil. And the reason behind this research is because the soil is a diverse ecosystem comprising living and non-living elements that interact in various ways. Soil is also concerned with nutrient cycling, pore creation, pore structure that are essential for soil health and soil quality. And it is the foundation of terrestrial ecosystem goods and services needed by human beings, but also with other living things. In this regard, soil health and soil quality are often used interchangeably. And in a natural and managed ecosystem, they are defined as the capacity of soil to function properly, to sustain productivity, to maintain plant and higher trophic levels, to maintain air and water in the soil. Um, Different regulators of soil function include, but are not limited to, the soil organic matter content, distribution of soil particle sizes, coarse soil fragments, soil acidity, density, water retention, cation exchange capacity, and so on. So in this regard, soil functions are constantly affected by climate change effects, particularly the provisioning, regulating, supporting, and cultural services. Impacts of climate change effects occur in different ways, typically through droughts, flooding, pest and diseases outbreaks, food stress, and heat waves. So these effects affect specifically and particularly low-income countries due to their climate patterns dependent economy. And this is because most low-income countries are depending on rain-fed agriculture, and variations in precipitation has a huge impact on food security, crop success, livestock production, and national economies. Further, in these countries, there is a continuous land degradation, reducing the potentials of the land to produce enough crop yield. If we take an example in sub-Saharan African countries, for example, global yields are highly depending on fluctuations of climate patterns, as agriculture is mostly rain-fed and crop management remains poor compared to developed countries. Land degradation related to climate change effects encompass changes in chemical, physical, and biological properties of the soil, which reduce the soil ability to sustain quality and quantity of plant growth, and hence the food security. climate change effects that have been reported to have a negative effects on soil properties include global changes in temperature and precipitation. These changes cause soil structure, changes in soil structure and soil porosity, changes in water infiltration, changes in root deepness, and so on. So the ecosystem-based approach, EBA, which consists of the use of biodiversity and ecosystem services to help people adapt to adverse effects of climate change, was developed as a solution to increase the resilience of communities to climate change. So it has been identified as a suitable approach to adapt and mitigate adverse effects of climate change on socio-ecological systems, and it has gained a significant consideration in different fields of environmental development policies. As a result, many projects have been implemented worldwide using ecosystem-based adaptation approach. In Rwanda, the approach was introduced to restore degraded wetlands, forest, and savanna ecosystem using agroforestry, bamboo, fruit trees, and indigenous tree species, providing cows and building rainwater harvesting tanks. So a recent assessment by different master students supported by REMA indicated that EB interventions was applied in 8 sites, and the restoration activities overall produced positive outcomes for communities through enhancement of direct ecosystem services and employment. However, despite the strong promotion and use of the ecosystem-based adaptation approach, building evidence of its environmental, social, and economic benefits in practice remains a work in progress. Currently, or right now, there is a lack of demonstrable evidence of ecosystem-based adaptation approaches in delivering ecosystem services, such as soil fertility. So, assessing effectiveness of ecosystem-based intervention to improve soil quality in restored lands using soil physicochemical properties is important but understudied in Rwanda. So there is a need to assess the contribution of ecosystem-based adaptation restoration activities on soil physicochemical properties. To fill the gap, the study assesses the levels of soil properties focusing on soil pH, soil electrical conductivity, ammonium, salts, nitrates, total nitrogen, organic carbon, available phosphorus, calcium, magnesium, potassium, sodium, cation exchange capacity, sand, silt, and clay soil structures, soil water content in eastern savanna and forest restored using ecosystem-based adaptation. The specific objective was to assess the levels of soil physicochemical properties in each restored land, and we have hypothesized that there is a significant difference in soil properties between the restored savanna and the forest lands. Of course, here I highlight that we did not be able to cover all possible soil physicochemical properties due to many and different reasons. The first one, the lack of materials to sample soil for the analysis of those other parameters, but also for the purpose of the cost, because the laboratory analysis of these properties is high. So this is the area of study. The study was conducted in 2 sites located in eastern part of Rwanda. The first one is located in the savannah in Nyagatare district. It is here on top. And the other one is located at Ibanda Makera forest, which is in Kirehe district. In each sampling site, 3 plots, here denoted by the letters A, B, C, have been selected, and the selection was based on how the land is affected by restoration activities, but also by other anthropogenic activities. Allow me to highlight that in Nyagatare it was very hard to find different types of lands because they all look similar. They are the lands that have been restored using different types of tree species, but cows continued to, to, to use the land where tree species have been planted. So the good different was at Kirehe district, where you have site A, B, and C, specifically A being located in a restored area, but on top of the hill, but also dominated by Lantana camara species. Then the plot B was located outside of the restored land, and it is dominated by the pasture land, and it has been selected to see if they can provide the outcomes that are similar to the pasture lands selected in Nyagatare district. Then the plot C was selected in a fully restored forest that is down at the low altitude and dominated by native tree species with complete canopy, showing really the good results of the restoration activities. So within each plot, 8 subplots have been selected, and each subplot had 50 meters, as indicated here on the figure. And within each subplot, 9 sampling points have been selected, and this gave a result of 24 soil samples in Savannah, here I mean in Nyagatare, and 24 soil samples at Ibanda Makera Forest, totaling 48 soil samples that have been taken to the laboratory of Rwanda Agriculture Board and analyzed for the indicated soil physical chemical properties in the previous slide. So in the laboratory, prior to the analysis, soil samples were received and they are dried and treated by removing stones, by removing the roots of some trees, and having the soil that is ready to be analyzed. And soil physical chemical properties were measured following the protocols in the laboratory methods of soil and plant analysis, where soil pH water was measured at the ratio of 2.51 water soil suspension. Electrical conductivity was measured by electrical conductivity meter. Total nitrogen was measured by calorimetric method through ultraviolet visible spectrophotometer. Ammonium and nitrate ions were measured by calorimetric method from a single soil sample. The ammonium of exchangeable sodium, potassium, calcium, and magnesium in the extract were measured by flame photometry, specifically for sodium and potassium, and by atomic absorption spectrophotometry for calcium and magnesium. Further, potassium, calcium, sodium, and magnesium elements were measured by complete oxidation of sample using Geldar procedures followed by spectrometric analysis. Organic carbon was determined using sulfuric acid and aqueous potassium bichromate mixture, and available phosphorus was measured by spectrophotometry at 884 nanometer. Then lastly, the water content was measured by the ratio of dry weight and initial wet weight of soil samples. The findings have been reported in an Excel sheet and then statistically analyzed using mean and standard deviation, but also by using the analysis of variance between plots and between the two sites. Results indicated that there is appreciable levels of soil pH in the lands restored in Kirehe plot C. And if I remind you, this is the plot that has been restored completely using native tree species and related it to the it is showing good results compared to other restored lands, while the plot C in Nyagatare also show good results in terms of pH water. And the explanation here is because it is at a lower altitude. I mean, both plot C at Kirehe and plot C in Nyagatare, they are at a lower altitude and they are nearby the river, and we suspect that plant heterogeneity controlled by water availability is the one control changes in soil physicochemical properties. The result about electrical conductivity indicated appreciable levels again in plot C, fully restored at Kirehe district, and the same result was also found in plot C at Nyagatare, and the same explanations as the one given for soil pH are the ones for electrical conductivity. We suspect water availability and plant heterogeneity to be driving these good results in terms of electrical conductivity. In relation with ammonium salts, here there is quite different because protein A and protein C at Nyagatare are showing the same results. The low levels were found in protein B, which is highly affected by cows based on high results in terms of ammonium salts were also found in plot B in Kirehe district. And if I remind you, plot B is located in the lands that are used for pasture land, and that are outside of restored lands in Kirehe. So if we look at the results at Nyagatare, specifically the levels of ammonium salts in plot C and plot A, they are almost similar for the ammonium salts in plot B at Kirehe district. So here we confirm that the effect of cows on soil properties focusing on ammonia seems to be the same at Nyagatare and in Kirehe district. In relation with nitrates, higher levels were found in plot C, which is nearby the river, but they were found in plot A in Kirehe, which is on top of the mountain, restored but highly dominated by Lantana Camara. And here, it's very hard to explain the reason behind these differences, but still, we can conclude that changes in properties and changes in concentrations might be higher for nitrates, particularly in disturbed areas, because even though the plot A in Kirehe is also stored, but it is disturbed by human activities, and the plant heterogeneity is not really good compared to plot C. In relation with total nitrogen, there are so many differences. There is no change, there is no variation at Nyagatare because all plots are affected by cows mainly, while it is higher in plot C at Kirehe, and here we suspect that this is due to the plant heterogeneity in the area, and some of them are leguminous with high ability to fix nitrogen. And this is related to the provision of good habitat for bacteria, nitrogen bacteria that are able to fix nitrogen. In relation with organic carbon, there is also complex findings. In Nyagatare, higher levels were found in plot A, and higher levels were found in plot C at Kirehe. And it is very hard to explain these differences, but we suspect that probably it's because the two areas have been restored using the same types of tree species. But here, deep analysis and deep investigation is needed to come up with tangible explanations of these differences. And if we go on available phosphorus, for available phosphorus, there is higher levels of phosphorus at Nyagatare, and then there is appreciable levels in Chirehe. When you say higher levels, that means they are the levels which are beyond of expected concentration of phosphorus in the field. But at Chirehe, the levels are appreciable, which is saying that restoration activities are contributing to the balance of soil nutrients. For calcium, there is also differences compared to the restored lands in savanna of Nyagatare and the forest of Chirehe. And there is really a big differences, because the higher levels in savanna found to be 7.6 MEQ, while in forest it is around 28.10. You see how there is these big differences. And once again, the higher levels of calcium were found in the forest restored using ecosystem-based adaptation. For magnesium, there is also the same trend. When you compare magnesium with calcium, we have found that higher levels are in plot A in Nyagatare and higher in plot C in Chirehe. So there are also some other differences, but when you compare the numbers, find that higher levels are available in Chirehe, especially in plot C, that is tight and away and showing positive results of restoration activities. The same trend was observed for potassium, sodium, and for cation exchange capacity. And there is no surprise, because cation exchange capacity is related to the positive ions that can be exchanged in the soil. So cation exchange capacity is corresponding to the changes in the elements that have been found in different plots restored using ecosystem-based adaptation. However, when it comes on soil structure, we found that the all plots in Nyagatare are dominated by sand soil, which is really high in terms of percentage compared with the percentage of sand soils found at Chirehe. And here the higher levels at Chirehe were found in plot A. And if you remember, this plot A is on the top of the hill, and we suspect that there is high erosion that is washing soil that is rich in nutrients, and the remaining soil is the one rich in sand. Another thing is that when there is the higher sand levels, there is a high level of infiltration, of water infiltration.
Okay.
Concerning the silt soil structure, higher levels were found in plot A in Nyagatare, and were found in plot B at Chirehe. And if I remind you, this plot is dominated by pastoral activities, mainly cows, and it is outside of restored land. So this similarity, higher levels in silt, might be related to the same land use in Nyagatare and Chirehe, but the differences in percentages might be dictated by the differences in altitude, because Chirehe is at lower altitude and Nyagatare is in higher altitude when you do the comparison between the two sites. The good thing is that Chilehe plots have uh, higher levels, especially plot C is higher levels of clay, and this might be controlled by two main factors: plant diversity, but also the location, because it is nearby water, nearby the river, and this might be related to high water content, as has been indicated by the result of this study. So if we do statistical analysis within plots, we found that higher levels of soil pH were found in plot C and in sward forest, which means that restoration provided improved soil characteristics because the level of acidity is reduced, but the plot had higher levels of electrical activity, total nitrogen, organic carbon, calcium, magnesium, oxygen exchange capacity, water content, and soil structure, which are the signs that restoration activities contributed to changes or to the standard levels of soil physical parameters compared with the findings in savanna, where there is a high levels of ammonium salts, but also higher levels of phosphorus, potassium, especially in plot A, while plot B, which is the neighbor of plot A, is rich in clay soil structure, then levels were higher
Sudah nunggu
in
ciko.
plot C
Agenda kuliah bukan.
located in savanna, nearby the river, nearby water, and it was sodium salt remained constant in all plots of restored savanna and forest. So in actually there is no change in the levels of sodium. However, a comparative study between plots located in savanna showed that there was no significant differences between sample medians of nitrate. This is the result from the analysis of variance, and this is specifically focusing on soil nitrates, available phosphorus, potassium, sand soil structure, electrical conductivity, and total nitrogen. Further, there was no significant differences in sample medians of nitrates, available phosphorus, potassium, sand soil structure, sodium, silt and clay soil structures between plots located in savanna and forests. So, besides the significant differences between sites, were found that there is significant differences in sodium, electrical conductivity, total nitrogen, and organic carbon. So findings of this study indicated that this data may serve as a reference for the future studies. So we can't conclude there is a change based on restoration activities due to the lack of baseline data. So these ones can provide as baseline for future studies while assessing the contribution of restoration using ecosystem-based adaptation to positive changes in the soil physical chemical properties. So far, most of the analyzed soil physical chemical properties are at appreciable levels if we go in the analysis of acceptable standards of the concentration in the soil physical chemical properties. So we recommend future studies to continue assessment of soil physical chemical properties after 2 years, 5 years, 6 years, and 10 years, and so on, to verify changes in the studied soil physical chemical properties. I thank you very much for kind attention and for following my presentation. If there is any support to improve this study, or if there is any question that you need clarification, you are welcome. Thank you very much.
Thank you so much, Dr. Venus. This is Diana Muton on the mic. Vena had to leave before the end of the presentation. So we appreciate that you set the baseline data for restoration soil properties, and I hope that we'll have so many researchers interested in this. So it was very interesting to see differences and similarities of parameters in Kirehe and Nyagatare, and to learn about the factors that were involved. So welcome to anyone who has a problem, suggestion, or any other input.
Thank you. Yes, Dominique, you are welcome.
Thank you, Dr.VENUSTE. Can you hear me?
Yes.
Okay. Yeah, thank you for your presentation. It's a very interesting presentation, interesting research. And I don't have a question actually to ask related to technical work you have done. But based on the technical laboratory related analysis you have done, I would like to know if you have a plan to extend to the northern side, because you were looking at some chemical properties, and if you have a willing or maybe a plan to extend to the north or western side of the country, so that we compare different parameters, climatical parameters or agroecological parameters between western, northern, and eastern side, because in my understanding, western and north, they have similarities in terms of topography, the natural environment, and then the eastern and some part of the south, they have also some similarities. So I wanted to know whether you have a plan for that, and then we see how we can look at this a bit of it. Thank you.
Thank you, Dominique, for the intervention. I wish I may continue doing such kind of research, but there is a limitation. This one has been funded by the University of Rwanda in collaboration with Rwanda Environment Management Authority and the Rwanda Higher Education Council. And for sure, the laboratory analysis is too high. So far, I don't have any additional fund to continue research in the northern part of Rwanda and in western Rwanda. But I agree with you that if it can be possible that we get enough fund, then we extend our research to other sites that can be more helpful to come up with the effect of restoration in different land use. But so far, I focused on this one where the restoration has been done using ecosystem-based adaptations, because the fund was limited on that. And I don't have extra fund to continue research in other areas. But my wish is that we may extend our study to other localities, especially in restored lands, to assess the effectiveness and the contribution of restoration activities on soil physicochemical properties. Thank you very much. I can see Rita Amanyire raised up the hand, and you are welcome for the intervention.
Okay, thank you so much, Dr. VENUSTE. My name is Rita Amanyira. I don't know if you can hear me.
Yes, I can hear you very well.
Thank you so much for the presentation. I'm from Uganda and I'm new to this field, but my question really is, how long did it take you to do this research and what inspired you to do it?
Thank you very much for the question. It took a long time because the sampling itself took around 2 months, because most of the areas are in a remote area like Kirehe. To reach there is very hard, and to sample in the area requires patience. So we had to spend 2 months on the field for sampling, and then we had to spend almost 3 months in the lab for analysis. And once we got the results, there's almost 2 months again for data analysis. So far, these are the findings we have, and we have been inspired to do this presentation to get the inputs and insights, maybe something to correct and some orientation, so that when we start writing the manuscript for publication, we will take into consideration um, your comments, your inputs. So, considering the inspiration, this is because I have been really, my area of research is ecology and sustainability, and I'm mainly focusing on soil lithotrophs, especially in different land uses. Here, the aim was to assess the diversity and abundance of soil lithotrophs in forest and savanna restored using ecosystem-based adaptation. Then I said, these issues of bioindicators is limited because most of the time people do not even understand. Why can't I include also a component about soil physical chemical properties, and then when I am done with biological data, I may link them and compare. Where we have good soil properties, is it where we have species rich, so species diversity of soil lithotrophs? So that is my inspiration. And the data we are presenting today is a component of a project that is in a way going, because I still have to analyze biological data, which so far I've collected, and we still have to run the laboratory analysis and come up with all statistical analysis to compare changes in self biodiversity in restored lands. That's how it came in my mind. I don't know if I satisfy your curiosity. Thank you.
Yes, you have. Thank you so much.
Thank you. Glorious, please come on. Yes, Glorious. I don't know. Glorious raised up the hand, but I can't hear her.
Oh, sorry, I was
In the
muted.
chat room.
I was muted.
Okay, come on, please.
Yeah, thank you.
Good, come on.
Thank you, Dr. Venuste, for your presentation. It was really interesting to hear about these results and different um, changes that happened. I have the same question as Dominique. I was also wondering if maybe in the future you will have to sample other areas. For example, I was wondering why your focus was only in Eastern Province. And my second question is about how you intend to disseminate this information. Like, for example, you want to publish this study in the future? Because I think if you can gather all stakeholders, for example, REMA, HEG, the Ministry of Environment, schools that are just having this kind of courses, maybe from these your recommendations, maybe other funds can come out to continue this kind of study.
Thank you very much, Glorious. I think the feedback I gave to Dominique is the one I'm going to give to you. That I'm relying on the fund. And the fund I had for this moment was limited. I could not go outside of the eastern part as the area of study I have indicated in the project. But my dream is to get all the data, soil data in restored land, let's compare the effectiveness of restoration. So the idea now came in my mind because before starting restoration activities, most of the time there is no baseline data. And it is very hard to assess the contribution of restoration activities. My dream, my intention was to do this research and provide data that can serve as a reference, that can serve as a baseline for future studies. This goes together to the question from Placide, who was asking, referring to your study results and what were expectations before a start, is your take now. So my intention was to provide the baseline data, and my intention is expand my research to other areas that have been restored, specifically using ecosystem-based adaptation and other kind of intervention. Then, this is the starting point of glorious research seminar. There shall also be an opportunity to do the presentation with the funder, and here I mean the University of Rwanda, Rwanda Environmental Management Authority, Higher Education Council, and when we talk about the Rwanda Environmental Management Authority, the Minister of Environment will be represented in this regard. And that's how we shall do the dissemination of our data here in Rwanda. And then we plan to do publication of this data to communicate the findings to the wide scientific audience working in the domain of soil sciences. Thank you.
Thank you, Venuste. Is it possible, for example, to invite you to give this seminar again at my institution? Because I think it can be one way of encouraging students to take part of this kind of studies, and also to be empowered, and also to, yeah.
I don't have any problem. Let us stay in touch. I may come
Yes.
and give the presentation. Thank you.
Thank you, Dr. Venuste.
Thank you so much, Dominique, Grace, and others for your input. So is there any other questions or any other input? Thank you so much, Salim. So Venus set the baseline. So I think it's up to us to go in and get more data, to provide those data so that we can understand if restoration really improves soil parameters. If you don't have any other question, I think you can end the meeting now.
I would like to thank you very much for your attendance, and we stay together for the next step. And